As we begin our conversation on C4 and CAM photosynthesis, it's really important that you have in your mind what C3 photosynthesis was. That's the original Calvin cycle. Why is it called C3? Because the very first thing that we make after we fix the carbon dioxide to the rubip is a 3-carbon molecule. Let me go back and give you the visual. So here you've got your carbon dioxide, you've got your rubip, you fixed it using Rubisco, and the very first thing you made had 3 carbons. This is what makes it C3 photosynthesis. When we start to talk about C4 photosynthesis, best guess on what the size of the first molecule is that we make? 4 carbons. The reason that I would ever want to do a different version of photosynthesis is that there's this process that happens called photorespiration. Photorespiration. What ends up happening is, if it gets really, really hot and the cell can no longer afford to open the underside of its leaf, imagine that stoma. Let's go back to the leaf picture. So remember that we've got our stomata. Stomata is the plural. Stoma is one. So we have these holes at the bottom side, and on either side of the hole are the guard cells. We talked about the fact that if guard cell is swollen, that means the hole's open, and if the guard cell is flaccid or limp, then the hole can close, right? So I have control over whether I have this hole open or not. Okay. If it starts to get too hot, for sure I want carbon dioxide coming in, and for sure at the same time, O2 is coming out, but so is water vapor. At the same time, as long as that hole is open, this is what's being exchanged. These are the gases that are being exchanged. As soon as I close that hole, no gases are being exchanged, which means that the CO2 stays outside and the H2O and the O2 stay inside, right? Because they are blocked from being able to leave. So far, so good? The problem is that the oxygen levels are going to build up. I'm going to get more and more and more, and I can't release the oxygen that I'm producing during photolysis. I'm splitting water, splitting water, splitting water. O2 builds up, builds up, builds up, builds up, builds up. And that would be okay, except that O2 is a competitive inhibitor. What does it competitively inhibit? It competitively inhibits Rubisco. So let's go back to the other cycle. If all goes according to plan, Rubisco has part of its active site where the rubip can fit. The other half of its active site is where the carbon dioxide fits. So imagine then that we've got ourselves an enzyme. This is just not the actual shape, but just an imaginary kind of a thing. Let's say I've got the active site where I can fit rubip in here, and then I can fit a CO2 over here, and then somehow that's part of my Rubisco. My Rubisco enzyme can do that, okay? The problem is that at that active site, sure, rubip can fit, sure, CO2 can fit. As O2 builds up, O2 builds up in here, it ends up getting in there first. And if it gets in there before the CO2 can get in there, then it's going to react with rubip instead of the CO2. But O2 doesn't have a carbon to give. So I'll end up breaking apart my rubip, but I'm not going to get that extra carbon in there. So here's what it ends up doing. So here's your key idea here. The oxygen competes with CO2. It is a competitive inhibitor, or at least competes for the active site of Rubisco. So we take our rubip, which is our 5-carbon molecule. When the oxygen gets in there, I split it into a 2-carbon molecule and a 3-carbon molecule. This one's fine. This is still 3-PGA, but there's only one of them, not 2 of them. One of these can go off to be a half sugar, but I needed 5 more. Well, it wouldn't exactly have been 3-PGA. I would have had to convert to the G3P, right? So that one G3P is going to go off to become a sugar. So maybe this guy can go contribute to that, but I don't have the other 5 that I'm going to need in order to make new rubip. So if I start doing this, I'm going to run out of rubip, and then everything stops. So I can have a slight sugar production, but not a great one. That means that I have effectively chopped down the rate of photosynthesis. Oxygen is interfering, and it's reducing the efficiency of photosynthesis. That's a problem for me. And that problem is called photorespiration. As opposed to carbon fixation, I'm not fixing carbon at all. I've been messed with. Okay? So this will happen. This happens this is very normal for plants. So under your conditions that you like, somewhere around 25 degrees is great for plants because the temperature is not too hot, it's not too cold. I've got lots of light during the day, so I can run the first part of photosynthesis and hopefully lots of carbon dioxide, no problem. But when it starts to get hotter and hotter and hotter, and I have to close the stoma, I have to close those holes so I don't lose water and dehydrate. If I close the stoma, I cannot get rid of oxygen. Oxygen builds up, photorespiration happens. Photorespiration is this oxygen here interfering with rubip so that I'm not producing the same number of half sugars that I wanted to produce back in the Calvin cycle. So it breaks my Calvin cycle down. Calvin cycle is useless now for the next little bit. Do you remember, though, that we said if you flood the system with lots of substrate, that you can overcome any inhibitors? This is why we said that. We were looking forward to this moment in time. If I have lots of O2 and I only have a little bit of carbon dioxide, because keep in mind, when that leaf is closed, there's no fresh carbon dioxide coming in either. Let's just draw that. So if I have the underside of my leaf here, okay, with the little stoma holes in the underside, normally I have O2 leaving and water leaving. This is normal. And then normally I have CO2 coming in, right? But when I have to close off the hole because I'm tired of losing water, the water stays inside, the O2 stays inside, and the CO2 stays outside. I cannot transfer them. So if I have CO2 stuck on the outside, I can't use it to actually fix it. It's not there. And I have all this O2 building up causing photorespiration. But if I can figure out a way, while I'm still able to open the stoma, to, as the carbon dioxide is coming in, store it, and have a huge pile of carbon dioxide already on the inside before I closed it, then I can release that carbon dioxide. There'll be so much carbon dioxide that the Rubisco will still be able to work because there'll be way more carbon dioxide available than the annoying oxygen that's building up. So I can still make sugar. I want to flood the system with as much carbon dioxide as possible. The only time I'm going to be able to get that carbon dioxide in there is when I'm able to open the hole in the other side of the leaf. So I'm just going to be smart about when I do that. So there's two things that are going to end up happening. The first one is called a C4 plant. C4 plants, these are the ones living in your hot, dry conditions. So things like grasses. I don't know if you know, but corn is just a fancy grass. It's a modified grass. It's just a giant modified grass. So is sugarcane. It's just a type of modified grass. It's just not like your lawn grass. Your lawn grass is the tiny version, right? So those types of ones tend to live in pretty dry conditions. Sometimes you got that poor corn sitting out in the hot, dry field for a long time. Same thing with your lawn. So it's quite a dry area unless you're watering it all the time. And that's why it tends to go brown. But what we're going to do is try to minimize the effect of photorespiration. So when it's hot and I have to close and I have to hold my breath because I don't want to lose water, then I'm going to increase the amount of CO2 I have available by storing it in something called mesophyll cells. Mesophyll cells. In a C4 plant, we're just going to have the CO2 will be stored or spatially separated, gets stored in one area so that the CO2 is constantly available. So here's what's going to end up happening. It's probably easier if we look down at the picture here. So this is our tubes, our xylem and our phloem. And right around, built into this area here, right around the actual tubes themselves, we have something called a bundle sheath cell. And then we have your normal mesophyll cells. And both these guys together are working to do the photosynthesis part. They're capturing the light, creating the sugar. Capturing the light, creating the sugar over and over and over again. Okay? Instead of the carbon dioxide being attached to rubip by Rubisco, instead we attach it to a different molecule, and it's called PEP, phosphoenol pyruvate. It's up here. PEP. Carbon dioxide gets fixed to PEP. It gets fixed to PEP instead of rubip. And because of the way that PEP is constructed, instead of making a couple 3-carbon molecules like the 3-PGA, which is what happened when we fixed the carbon dioxide to the rubip. Instead, what I'm going to get is a 4-carbon molecule called oxaloacetate, and then malate will be formed after that. Oxaloacetate is a 4-carbon molecule, not a 3-carbon molecule. That's why this is called C4 photosynthesis. Let me say it one more time. Calvin cycle, CO2 plus rubip makes 3-PGA. It's a 3-carbon molecule. In this C4, carbon dioxide plus phosphoenolpyruvate plus PEP produces oxaloacetate, a 4-carbon molecule, which then gets converted to malate, and then the malate gets shuttled into this other specialized cell, this bundle sheath cell, and then I just chop off the CO2 again. Once I chop off the CO2, malate minus the CO2, so 4-carbon, minus the 1-carbon, gives me back this pyruvate molecule, which is a 3-carbon molecule. I go back to making PEP by just adding this phosphate group. Phosphoenolpyruvate is how I've attached the phosphate group to the pyruvate molecule. So I'm just add that on. The PEP carboxylase is the enzyme that's carboxylating the PEP, make the oxaloacetate, make the malate, come back around to make the pyruvate, and just keep doing that. And what I'm doing is I'm giving myself a chance to take this carbon dioxide and store it down here. So I've been doing this the whole time that I've had the hole open. I've been storing. So I have a ton of CO2 down in this bundle sheath cell. And what's special about these cells, is why we care, is that these cells, their cell membranes are impermeable to CO2. Once I put the CO2 in there, it's stuck in there, which is great. It's like a container that I won't lose CO2 from. So as long as I'm throwing CO2 into this container, I'm good to go. So up here, I can perform the first part of photosynthesis, where I'm producing oxygen like crazy. No problem. That's fine. Let the oxygen build up, because I've got all this carbon dioxide down here, and this is where I'm going to use my Rubisco, in this separate cell that doesn't have the oxygen building up. And so because there's so much CO2 available and all the oxygen is being built up in this cell in a different place, I can run my Calvin cycle like normal. So here's your key. C4 plants are going to store the carbon dioxide in malate molecules in a separate bundle sheath cell. And they do that by fixing the carbon dioxide to a PEP, or a phosphoenolpyruvate, rather than rubip, using PEP carboxylase instead of Rubisco. And then they can run the Calvin cycle like normal. That means Calvin cycle is Rubisco, right? That means that every plant can still do Calvin cycle. Every plant can do Rubisco. These guys just have an added step to overcome the constant problems with heat, constant problems with water loss and transpiration from the tissue. I hope that makes some sense. I can go over that a million times if you like, but that's the general idea. C4 plant, special spatial separation, do all of the light reactions here and let the oxygen build up, do all of the Calvin cycle stuff in a separate place, so I don't have to worry about photorespiration. So I can use Rubisco without being inhibited. Last conversation then is, that's C4, what the heck is a CAM plant? We're going to see a very similar process happen here. CAM plants are the type of plants that live in extremely dry conditions. It's not just a moment in time when they're like, oh, it's a little hot today. It's hot every day. So these are your desert or your very dry conditions. CAM plants then, these are the type of plants that you would normally call your cactus or things like pineapples. They're called succulents. They're called succulents because they are storing so much water in their actual bodies themselves. So these are your fat, like your typical cactus that you would see. They have the same chemical pathway as C4. I'm going to store the CO2 as the 4-carbon oxaloacetate, which becomes malate, which then gets knocked off, which leads to the carbon dioxide. The difference will be I don't have a special bundle sheath cell. So it's not that I have the light reactions happening in one place, in one type of cell, in the mesophyll, at the same time that it's happening in the other one. Instead, what I have is just separate the timing. One cell, but one part of the day you'll be doing the light reaction, and then the other part of the day you'll be exchanging gases. And of course, you're going to exchange the gases when it's cool at night. So the timing is different, not the space being different. So let's look at the side-by-side pictures. So on this side, we have C4. Grab your carbon dioxide, fix it to PEP, eventually make malate, transport the malate into this special cell, knock the carbon dioxide off, run Calvin cycle like normal, I can make my sugar. So the light reactions happen up here, no problem. Oxygen can build up, I don't care because I got so much carbon dioxide stored down here, I can still run Rubisco. So this is Rubisco down here. This is PEP carboxylase, this is Rubisco. For the CAM plant, all I do is, because I can only open the holes at night, CO2 comes in at night, no problem. I'm sucking it in all night, storing it as malate. And then during the day when I'm forced to close my stoma because I don't want to lose all that water, then I can still run the Calvin cycle because I have flooded the area with this CO2 substrate. And that's the end of our conversation for C4 and CAM plants.